Hello and welcome to today's lecture of Introduction to Mechanobiology. So, in the last class we had started discussing about differences in the type of focal adhesions that you might observe in different cells and what drives their assembly. Okay. So, in that context I had said that there are three different types of focal adhesions, the smallest being focal complexes. Okay. These are more dot like, order 1 micron in length. They are present at the edge of the lamellipodia. And these are typically short lived. And in terms of composition, they have focal adhesion proteins like paxillin and vinculin present in them. Okay. So, from focal complexes, these focal complexes mature into larger focal adhesions. Okay. So, focal adhesions are more elongated in shape. These are roughly 2 to 5 microns in length. Okay. And they are present at the cell periphery. And in addition to paxillin and vinculin, so of course, integrins are always there. You have many more proteins including tailin and alpha actinin. Okay, so this pen is not working. Okay. So, these focal adhesions are present at the cell periphery. So, from focal adhesions, in addition, you can have fibrillar adhesions. Okay. So, as the name suggests, they are fibrillar in nature, but they are present centrally, okay, towards the center of the cell. They can be 1 to 10 microns in length. Okay. And in addition to integrin, they have this molecule called tensin. Okay. So, if I were to draw a schematic of the cell, okay, your focal adhesions are present here, okay, and the fibrillar adhesions are present here. Focal adhesions are also present at the rear of the cell, and you have focal complexes. Okay. These appear like dots, they are really small in size. So, you have focal complexes mature into focal adhesions at the cell periphery, and these become fibrillar adhesions at the cell center. Okay. So, what drives this transition? It is force. So, force drives maturation of focal adhesions. Okay. So, this requires force, and one of the first studies which demonstrated that was by the group of Sasha Bershetsky at Weizmann. Okay. So, what he showed was by exerting force. So, you have a cell here sitting on a substrate connected by adhesions, and he took a pipette, rolled it on the cell edge, and he showed that. So, in top view you can see that wherever you put the pipette, next to this you see growth of focal adhesions. Okay. So, this was done with GFP vinculin transfected fibroblast. Okay. So, this paper I have also recommended you to read as a reading assignment. You can download it through PubMed. Okay. So, however, though this experiment demonstrated that forces are required for driving the growth of focal adhesions, it is difficult to quantify the forces. Okay. So, one more thing that this study demonstrated was this force dependent growth occurs at integrin additions, at integrin based additions. Okay. So, if you were to do the same experiment on a polylysine coated surface, you will not see it. Okay. So, the problem with this particular experiment, not the problem, the caveat is that you cannot measure the forces directly. So, this called for an alternate approach or technique to, so that you can quantify these forces. So, one of the approaches which was developed by Benny Geiger and Sasha Borshetsky at Weizmann was, what they did was they took a substrate, a patterned elastomer, where they coated fibronectin spots on top of this elastomeric substrate. So, this elastomer was PDMS. Okay. So, the grid like location spacing of these dots will allow you to quantify how much one of these dots get displaced when cells are sitting on that, and by multiplying this by the local force, you know what is the force exerted at that particular point. Okay. So, through this experiment, what they found was, if you take a cell, okay, you have these adhesions, okay. So, these focal adhesions, and at each focal adhesion they measured, they were able to measure the forces. Okay. And what they found was that there was a linear correlation. Okay. So, there was a linear correlation between the force that they measured at a given addition and the area of this addition or the size of this addition. So, this was roughly linear. Okay. So, this was a linear dependence, suggesting that there is a correlation between force and area, okay, force and focal adhesion size. Okay. So, not only the correlation holds between force and focal adhesion size, if you were to track the intensities, because this experiment was done with GFP transfected cells, GFP vinculin transfected cells, you can track the total intensity of a given focal adhesion and the force measured at that point. Once again, you have this linear correlation. Okay. Size and force and intensity and force, which means that given if a focal adhesion is bigger, there is increased amount of localization of that protein at this adhesion. Moreover, so the way I drew this picture, I roughly aligned the direction of the force and the major axis of the focal adhesion. Okay. So, if you were to look at the force orientation, and the focal adhesion orientation. So, by fitting an ellipse to each of the focal adhesions, you can find out the angle of the major axis, and they found was even there was some correlation. So, there was kind of a linear correlation. Okay. So, still this establishes an association between force and focal adhesion size, but it does not say what is the directionality of this association. Okay. So, to demonstrate that or to test that, what they did was they did experiments in the presence of this drug called BDM. So, what does this drug BDM do? It relaxes cells, or in other words, cells exert lesser forces. Okay. So, you are perturbing the magnitude of the forces which the cells were exerting. Okay. So, what they observed by doing this experiment, so if I were to draw this curve again between force and area, FA area, okay, the nature of the curves remain the same. Okay. So, if earlier points were up here, in the presence of drug, these points shifted to the left. but they fell on the same line. Okay. And on top of that, you could find out, okay, a force. So, this line has a non-zero intercept. Okay. So, has a non-zero intercept, and you can also find out the slope of this line. The slope of this line was roughly 5.5 nanonewton per micron square. So, this was the average force sustained by a focal adhesion of size 1 micron square. So, 1 micron square focal adhesion sustains a force of 5.5 nanonewton. Okay. So, and if when they did the experiment of trying to see as a function of time, okay, how the force dropped. Okay. So, when you administered the drug, let us say this is time 0 when the BDM drug has been added. Okay. So, before that point you have some constant value, and then these values after beyond this transition point they start to drop. Okay. So, correlated with this particular drop in focal adhesion force, when they also plotted the focal adhesion, the total intensity, okay, this also exhibited a drop. Okay. So, this is for this axis and the blue is for this red axis. Okay. So, once again, so concomitant with drop in force, there was a linear drop in the total intensity of the focal adhesion, and also, so these were correlated, perfectly correlated. So, this experiment suggests that tension or force exerted by cells at focal adhesions actually stabilize the focal adhesion. Okay. So, force is required to stabilize focal adhesions. Okay. So, here you see, so again if I were to go back to my analogy of cell as a tensed rope or tensed rubber band, right. So, imagine a simplified picture where, okay, so this is my substrate, this is my theoretical cell, okay, which is like a tensed rubber band. Okay, and these blue represent my focal adhesion. Okay. So, the more amount of increase, so if you increase the tension, so I can plot. So, if the tension or force exerted at the, tension or force exerted at the adhesion versus the substrate compression. Okay. So, you should see a linear relationship. So, the more the tension, and if the focal adhesions are stabilized, then the substrate will get compressed, and not just substrate compression, you can say along with this the FA size will also increase. So, that is how this entire system is stabilized. So, increase in tension allows stabilization of focal adhesions, the entire structure gets stabilized, and that is how your, and the substrate, the forces get transmitted from the cell to the substrate. So, that is why you get increased substrate compression. Okay. So, this brings to an end our discussion on focal adhesions. I would once again give reading assignment of the two papers that I discussed over the course of the today's lecture and last lecture. Okay. So, these are the two papers you must read, you can download it. Okay. So, you can download from PubMed. First paper is Rivlin et al. Journal of Cell Biology 2001. The second paper is Balaban et al. Nature Cell Biology, again 2001. So, these were two landmark papers which demonstrated the influence of forces in regulating focal adhesion size. Okay. So, coming back to our analogy of cell as a tent. So, we have discussed the ECM, we have discussed focal adhesions. Okay. Now, we will come to the cytoskeleton, right. So, the cytoskeleton is the machinery which is required for cell stability, okay, but also this is the one which regulates the forces which are acting at focal adhesions. Okay. So, for the most of you know what cytoskeleton is, but I still thought of giving a brief overview of the cytoskeleton. Okay. So, there are three different filament types, okay. One made of actin, one made of microtubules, one made of intermediate filaments. Okay. So, today, so the overall properties of the cytoskeleton. So, though there are three different networks, but there are proteins which cross link or link actin and the microtubule cytoskeletal. One example is this protein called ACF7 or actin cross linking factor 7. Similarly, you have proteins which link the actin to the intermediate filaments and the microtubules to the intermediate filaments. So, interactions between the three filament types determine the mechanical behavior of the cell. Okay. So, today we will get started with discussing actin. Okay. So, actin is actually one of the most abundant proteins. Okay. So, one of the most abundant proteins. So, roughly 10% of the total protein in eukaryotes is made of actin. Okay. So, this amounts to roughly 500 million molecules. Okay. So, if you look at an adherent cell, so if I were to draw an adherent cell, let us say if I draw a cell like this and I look at the actin cytoskeleton, you might have as well as. So, I have drawn these peripheral stress fibers. So, this is called the cortical actin network. Okay. So, these ones are called transverse dorsal arcs. Okay. These ones are called dorsal stress fibers. Okay. So, in addition to forming these fibers, you will have a diffuse background. So, these are referred to as a network contraction array. Okay. So, you have various different actin structures. If I were to draw the cell of a migrating cell, okay, if I were to draw a migrating cell, so you can see branches which form. Okay, you will have monomers which will polymerize against the membrane and push it. You will have these stress fibers as well. So, this some of these, so at the leading edge of the cell, these are more dynamic and these are more static. Okay, so, these are the ones which actually transfer forces to the substrate. Okay. So, there are two pools of actin, or actin exists in two forms. Okay, I have F actin and G actin. Okay. So, G actin stands for globular actin and F actin stands for polymerization, filamentous actin. Okay. So, your G actin monomers, okay, so come together to form a filament. So, F actin is just, okay, formed by polymerization of G actin. Okay. So, what has been demonstrated by a range of studies by lot of researchers, that if you take monomers, okay, and these apply for multiple different type of processes. Okay. So, what you first need is the formation of a nucleus, okay, for some of these monomers, and then spontaneously these nuclei assemble in straight line, and then you have these monomers getting added and, okay. So, these monomers then come. So, this entity is called a nucleus. Okay. So, these green dots are my G actin and this is my filament forming. So, this filament, in this filament you can have a new monomer getting added or coming out of it. Okay. Similarly, you can have addition or removal from one end. Okay. So, in steady state, okay, your filament length will roughly remain constant, but it is dynamic, right. The process of addition, so process of addition is polymerization or removal, which is depolymerization, will continue. Okay. So, if I were to plot it in terms of time, so it takes quite some time for these monomers to come together from a nucleus, and then once the nucleus is formed, you have quick formation of a filament. Okay. So, my y axis is the mass of filament or filament content. But if I provide a nucleus, then this process is much hastened. Okay. So, this is when you add a nucleus. Now, this filament formation does not happen at all times, but will require a critical concentration. Okay. So, if I plot the monomer concentration, okay, so below a certain threshold, okay, below a certain threshold, you will have, if you keep on adding monomers, your mass of the monomers will keep increasing. But once this threshold is crossed, okay, your monomer concentration will remain constant, but your filament concentration will increase. Okay. So, this is your filament and this is your monomer. Okay. So, this is a critical concentration. So, Cc, so this represents the concentration of unassembled monomers at steady state. Okay. And this has been found to be 0.1 micromolar. Okay. So, in next class we will continue from here and discuss how actin filaments grow, what kind of network you can form and how can you measure the forces exerted by these actin networks. Thank you for your attention.